Hi, my name's Victoria Schlesinger. Welcome to Bay Nature Talks. I'm the editor of Bay Nature. If you're not familiar with Bay Nature, we are a magazine and website, nonprofit independent reporters who cover the environment in the San Francisco Bay Area. We have been publishing for 20 years. And 10 days ago, we published this, our fall issue of Bay Nature. You can go to baynature.org and subscribe, or go to baynature.org and you can read anything in this magazine or anything we've ever published for free and donate. We are small, we rely on donations, so thank you. And extra thank you to those of you who gave the $20 suggested donation for today's talk. We really appreciate it. Today, we will be talking with two people, Elena Konis and Daniel Roman, about their reporting for a major feature in the fall issue of Bay Nature called Epizootic, how infectious disease goes from, you gotta see this great artwork, goes from wildlife to humans and back to wildlife. But before we get to talking with our guests, I want to go through just a little bit of housekeeping. We're going to talk, Elena Roman and myself are going to talk for about 35, 40 minutes, and then we'll go to a 15-minute Q&A session. You can use the Q&A function at the bottom of your screen to ask questions at any time during our talk, and then I will get to them all, or as many as I can at the end, and we'll go through them then. Please don't use that Q&A function for technical questions. If you're having trouble, we're so sorry, we're not really equipped to help you. If for some reason your technical issues keep you from participating and watching today's talk, you will get an email if you've registered for this event with a link to the event, and you can watch it later. So without further ado, let me introduce our guests. Elena Konis, who is a historian who specializes in the history of public health. She's a professor at UC Berkeley, and she teaches both in the graduate journalism school and in the School of Public Health. She's written about infectious disease quite a lot, and is the primary author on this story we're going to be talking about. I'd also like to welcome Daniel Roman, who supported Elena's reporting for this story. Daniel is a second-year master's student in the UC Berkeley Graduate School of Journalism program. And prior to journalism school, he did some very cool work for 4 years at Oakland's Youth Alive, which is an organization I've just learned about. That sounds great. So let's see. Welcome, both of you.
Thank you, Victoria. Thanks for having us today.
Yeah, it's a pleasure. I'm excited to talk about this story, and because it is such a story, it's such a yarn, and the way you've written and reported it, it's really a page turner, and one that is very apropos to our times when we are experiencing things for the first time in our lives, and sometimes can feel that they are the first time ever. Even this last week, in some ways, feels unprecedented.
Yes.
But we know when we look to history that is not necessarily the case. So would you, Elena, set us up with a little bit of what was the world like, what was the US like, San Francisco like, when the first pandemic hit the US maybe 100 years ago?
Yeah, thank you for that question, Victoria. And as you mentioned, I'm a historian of public health. Specifically, I focus on the history of infectious diseases. And one of the joys and one of the perils of being a historian is that you have all of history to think about. But I just want to confine my answer to your question by thinking about 100 years ago, because 120 years ago, rather, is when plague, the disease that Daniel and I wrote about for the current Bay Nature issue, that's when it first arrived in the US, coming through the port of San Francisco. And these days, I think, the year 2019 and the year 2020 now have had a tendency to make us feel as if we're living in really unique times, like you mentioned. Like we have more infectious disease threats than ever in just recent years: measles, flu, now COVID-19, something that has really brought us to our knees and paralyzed us as a society, make us feel as if this is absolutely unprecedented. But it's absolutely not. And if we rewind the clock 120 years ago to around the year 1900, things like quarantine were a really common occurrence. They happened every time somebody came down with a case of measles, a case of mumps, a case of scarlet fever, a case of chickenpox. At that time, we were just seeing the first few cases of polio. And I mention these diseases just as a reminder that at that time, infectious diseases were so common, and they were a routine part of childhood in particular, but also of teenage years and adult life too. And over the course of the 20th century, we started to see a number of those diseases decline. First, just due to better standards of living and better nutrition, better infrastructure. But in the 1950s, they started to decline due to antibiotics and then vaccines, really good vaccines that we started to develop, especially in the later 50s and 60s. And this was such a dramatic shift that by the mid 1960s, infectious disease specialists were predicting that we were about to see the end of infectious disease. They were actually predicting a future in which we were just going to live free of them. Today, that seems absurd, but back then, experts, scientists really believed that. And really, from the 60s, 70s until the early 80s, we had a lot of reason to think that we were headed in that direction. It changed with HIV/AIDS. And then, since the 1980s, we've lived in an era of kind of emerging infectious diseases. And you could add to the list Ebola, West Nile virus, the first SARS, and now COVID-19. So I see us in a way as almost returning to a state that we were living in the past, when infectious diseases were very, very common and very, very debilitating. So it's an uncomfortable thing to say as a historian, but in many ways, this moment feels familiar. It's nothing I ever thought I'd live through, but it feels familiar from what I've studied and what I know of the past.
So can you take us to the story that you tell in Bay Nature about the arrival of plague to San Francisco? It's really dramatic.
Yeah, I think I'll let Daniel start because it kind of came in two waves, and he can describe the first one and I'll describe the second one.
Okay, great.
Yeah. So, at this point, plague had a long history, right? It had been around for more than 1,000 years. People mostly knew of it because of its association with the Black Death in Europe in the 14th century. But what happened was, around the mid 1800s, a series of plague outbreaks had started in southern China. And basically, by the late 1800s, plague was appearing and moving from one Pacific port to another. So some coastal cities it was hitting included like Hong Kong, where a plague outbreak there killed 2,500 people. Another city, Canton, which is just northwest from Hong Kong, an outbreak there killed more than 35,000 people. So plague was spreading, and it was really just spreading along like heavily trafficked trade routes, hitting other places in China, but also spreading out to major port cities in places like India in the Middle East. So, at this point, it was really only a matter of time before, plague showed up in a US port. And in early 1900, that's exactly what happened. So for San Francisco, it really all started with rats. Rats that probably climbed aboard a ship that was docked at a plague-infected city before arriving in San Francisco. That plague-infected city was probably Honolulu, which had its own plague outbreak just a couple months before San Francisco saw its first case. So once this ship docked at San Francisco, these plague-infected rats found their way from the pier and up into Chinatown. And, the rats likely gained entrance into this specific neighborhood, Chinatown, through sewer pipes, because Chinatown sewer pipes emptied at the port of San Francisco. So this is likely where the rats gained entrance. And this is also probably why Chinatown was hit first. Well, one of the reasons why. But anyway, by March of 1900, San Francisco sees its first case with a man named Wong Chuk King, who is the first plague victim that the city sees. So, at this time, it should be noted that at this time, the physical environment of San Francisco in general was a very hospitable place for rats. On top of already having a large rat population, San Francisco in general had poor sanitation, poor waste management, and everything was made of wood, wooden houses, wooden decks, wooden sidewalks, wooden stagecoaches, etc. So this part about wood was pretty important because it was perfect for rats. It enabled the rats to kind of slip through and hide, dig through, and sort of like nest within these wooden structures. So in terms of just San Francisco being a hospitable place for rats, this was no different for Chinatown. And if anything, living conditions were probably a bit worse for residents in Chinatown. And that's just because, if we look at the history, due to discrimination, Chinese people were not allowed to live anywhere else in San Francisco besides Chinatown. So, at the time, Chinatown, the neighborhood itself just consisted of a tiny, 12-block radius and was home to almost 30,000 people, and that was growing. So when you have that many, yeah, so when you have that many people in that small amount of space, you get, overcrowding, a lot of shared living spaces, overall cramped living conditions. So that's how it was for Wong Chuk King. He shared a bed with three other people in a cheap hotel in a basement, right next to an underground cesspool. And there was an open sewer pipe that ran into the room. So all these conditions just made it easy for the rats to flourish, multiply, spread, and thus this made it easy for plague to spread, because it was being transferred by fleas.
Right. I was going to say, can you just explain how that transfer works real quick in case some people don't know?
I can actually explain it, and maybe Daniel, you can bring up one of the photos that we have of,
Sure.
of living quarters. And for plague to spread, it takes a rat or a rodent, and it takes a bacterium called a bacillus, and it takes a flea to take a bite from the rat, get the bacillus into its body, and then bite a subsequent rat and give the bacillus or the bacterium to the next rat. And that's how plague spreads among rats. It spreads to people when one of those fleas jumps from a rat to a person and takes a bite of a person. And inside the flea's body, that bacillus multiplies and multiplies. It forms these clumps of bacteria that then actually block the flea's foregut. And when it takes a bite of, let's say, a person, it just regurgitates all of that bacteria, which then enters the person's body. And when rats and people are close together, the chances for the flea to move to the person increase accordingly. And I'll let Daniel describe this photo of the time.
Great.
Yeah, so this photo right here is an example of a living space that you might find in Chinatown at the time. So this is a photo of basement living quarters in Chinatown. As you can see, the floor, the deck right there is made of wood. You can see there's spreads for a couple people that are probably occupying these spaces. So yeah, so this is a typical example you'd find in Chinatown at the time. Let's see, I have another photo. Let's see. Sorry, let me just, there we go. So this photo right here, so during this first epidemic in San Francisco, which lasts from 1900 to 1904, basically what was done to kind of control the spread of plague was health officials under the guidance of Joseph Kenyon and then Rupert Blue who were officers in the medical, or excuse me, Marine Service, the Marine Hospital Service Organization. They were sort of in charge of overlooking and sort of stopping the spread of plague in San Francisco. So one of the things they did was they would tear down, rat infested buildings, or tear down buildings, mostly wooden buildings that were suspected of harboring rats. So this photo right here is an example of that. This is some public health workers tearing down a wooden structure that was suspected of harboring rats.
And at that stage, was plague in San Francisco primarily still confined to Chinatown, or had it spread out more widely? Or
it was confined to Chinatown for the most part. So the total of cases for this first epidemic was about 121 cases, and most of those were residents from Chinatown.
who are clearly losing the housing they desperately needed.
Right.
Yeah, and the other piece of the health officials' response to the disease at that time was to quarantine Chinatown. And effectively, by quarantining it or cordoning it off, is the term that they used at the time, they effectively kept the disease contained there, but they used pretty racist policies and procedures to do so. They let whites cross the cordon and focused all of their efforts on the Chinese American residents living in that district. Interestingly, after 4 years of these kinds of efforts, destruction of wooden structures and isolation and quarantine of affected largely Chinese American residents, plague disappeared, and then it came back after the 1906 quake. And after the 1906 quake, which hit San Francisco in April of that year, it destroyed 80% of the city's buildings. Everything was just destroyed. Fire followed. And because the city's water lines were broken, the fires just raged. And you had hundreds of thousands of people living in refugee camps and tent encampments on the streets, and nowhere for the rats to go but into those encampments. And so it was only a matter of time. And sure enough, by 1907, another case of plague appeared and a second epidemic materialized. And this one spread throughout pretty much every residence or residential area in the city. It affected every neighborhood. It affected people of all races. And the city took a very different approach. It had less destruction to do because the earthquake and the fire sadly had done that, but they rebuilt the city out of concrete. Anything that had once been made of wood now had to be made of concrete. Sidewalks, wharves, docks, piers, the bases of chicken coops back in 1907. If you wanted to build a coop, you had to lay down 2 inches of concrete first.
Sure.
They also paid the citizens of San Francisco to bring every rat they could find to the health department. So 10 cents a rat was their rate. And they autopsied every single rat that was brought in. They brought them in to see if they had plague, and they kept paying people until they believed that they had found the very last rat with the plague bacillus in its body. And I think we have one more picture. Daniel, if you could go forward. This is them preparing rat bait. Rat traps were set all over the city with all kinds of tasty bait. And then people would lay out those traps. Neighborhood organizations and school kids would lay out those traps and then collect the rats. And health officials, I think this is in our next photo, then cut the rats open and check to see if there was any bacillus left in their body.
Wow.
And by 1908, they were confident that they had found the last human case and the last rat, and believed that they had completely rebuilt the environment in San Francisco to make it far less hospitable to rats. And in fact, plague stayed away. Of course, there's more to the story, but
Yeah. So that, I mean,
I'll hold right there for now.
For people who are listening who know Bay Nature, I mean, the reason Bay Nature ran a story about this is because plague has a whole second life that Elena will tell us about, that is fascinating, that I really wasn't as aware of as I am now, this whole syllabic cycle to it. So why don't you tell us how how that happens.
Yeah, I think Daniel, you can probably say a few words about the first few kind of moments when people realized it had spread beyond the rats and people of San Francisco.
Right.
Yeah. So in 1903, health officials in San Francisco started noticing like the arrival of hospital patients from the East Bay who had plague. So these people were coming from areas that that didn't necessarily have a plague epidemic like in San Francisco. So there was a case from a person from Pacheco came into with plague. A person from San Ramon had plague that came into the hospital. So
And so different back then. I mean, whereas now we feel like, oh, I'll just go to San Francisco. It was a little more complicated back then. You had to get in a boat. You had to take a real journey. So,
right.
And those were rural areas. They were really
Very.
rural areas then.
Yeah.
Right. Yeah. So, up until this point, plague cases were mostly, in San Francisco. So, this raised suspicion, like, why are these people who are not from San Francisco, why are they getting plague? So Rupert Blue, who by now had replaced Joseph Kenyon, started to investigate. And so, he makes a trip to the East Bay, and he basically learns that, for one of the cases, specifically for the person from Pacheco, he learned that prior to becoming ill, this person had shot and collected like several local ground squirrels, prior to getting sick. And then Blue found something similar for the other person who came from San Ramon. This particular person was working at a railroad camp, and basically, you know, the workers at the railroad camp were known to kill squirrels and eat them. You treat them as food.
Yeah.
So this obviously, Rupert Blue raises even more suspicion. He sees the connection, and so he sends a physician later on. He sends a physician into a couple East Bay counties to do some more investigation. And sure enough, the locals informed this physician that, how like in places where ground squirrels were pretty common, they were noticing that they were disappearing. And another local had told a physician that, how he heard that other ranchers were bringing back dead squirrels to their land in hopes that whatever sickness they had, they would spread it to the squirrels in the area. So
right. And ground squirrels, as Bay Nature's reported on a lot in the last year, you have a long contentious relationship with farmers because they build these elaborate networks of tunnels and really disrupt farmers. So many people in agriculture would really rather not have them around. But
right.
yeah,
Right.
go on.
Yeah. But yeah, so ultimately, after that trip with the physician, ultimately Rupert Blue ends up sending a team of people to collect rodents in Contra Costa County to test them. And out of the more than 500 animals that they caught, few were infected with plague, but nonetheless, the conclusion was that plague had indeed made its way into ground squirrels of the East Bay. And so it's in 1903 where health officials first see any sort of indication that plague had indeed entered the wild rodent population in the East Bay.
Yeah.
And what's interesting about this is that until that point, plague was considered just an urban disease. And now plague experts, and I think they first kind of outlined this more than 50 years ago, they outlined sort of three eras of plague in the US. And in this first era, plague, despite the discovery that it was in ground squirrels in the East Bay, it was still thought of as an urban disease. And for the next 20 years, from the 1900s to the 1910s into the 1920s, plague showed up at ports on the West Coast, ports in the Gulf, almost never through ports along the East Coast where it was just too cold. But plague was considered a disease that you only had to worry about if you were a human, if you were living in a city. And now scientists talk about two different cycles of plague, the urban cycle and the sylvatic cycle. So what happens after this period of urban outbreaks, which I think the last one was in Los Angeles in 1924, it's followed by a period from the 1920s to the 1960s When plague stops appearing in people, there are remarkably few cases in people, but it starts spreading among the wildlife of the West. And people start reporting ground squirrel die-offs in multiple parts of California, then up in Oregon, then over in Nevada, then prairie dog die-offs in New Mexico and Arizona, and then Montana and Idaho start to see die-offs of rodents. And by the 1930s, the Surgeon General, Thomas Perrin, decides this is worth investigating. And he sends two of his researchers, two physicians, Dr. Haas and Dr. Eske, out to the Western states. And he asks them to kind of figure out how far and wide has plague spread among the wildlife. And so they outfit this Jeep. And I think, Daniel, we've got a photo of it if you don't mind bringing it up.
Yeah.
And in the late 1930s, they drive across the Western states, pretty much everything west of the Rockies. And they're collecting all kinds of animals, chipmunks, what they call native mice and rats, groundhogs, which they admit are inaccurately named, prairie dogs, marmots, ferrets, and they bring them back to their truck and do essentially what the San Francisco Health Department had done. They cut them open and look to see if they have signs of plague. And after several years of doing this, they report back to the Surgeon General that they have found plague in 11 Western states. And they write this almost poetic sentence. They say that plague now exists from seacoast to mountain, from desert to forest, from valley to plateau. A great reservoir of infection has become established in the US West. And you can see them, and sometimes they worked with local health officers too, working out of the back of their truck here. So what they discover is that, everybody celebrated that plague had been controlled in the cities and sort of forgot to keep track of what it was doing in nature. And what it was doing in nature was absolutely devastating, causing, and this goes to the title of the piece in Bay Nature, causing epizootics, these epidemics of the disease among wildlife. And some of them were really dramatic. Still, at this time, though, ranchers had no problem with it. They probably had the closest view of plague, but there are reports of some ranchers driving hundreds and hundreds of miles to try to find a sick or dying prairie dog, for instance, to bring it back to their ranch so it could kill off all the prairie dogs, infesting, as they thought of it, their land. So agriculture probably had a hand in in plague spread, but also plague was what was becoming known as a generalist. It could infect all of these species, and so it did.
And continues to.
Yeah.
You wrote in the piece about some current examples, like the black-footed ferret, I believe it was. Could you talk about that and vaccination?
Yeah. So today, and I think probably Daniel has these numbers at his fingertips, but I mentioned before that there are three eras of plague. The first one, it was a largely urban disease. In the second era, from the 20s to the 60s, it was an almost exclusively rural wildlife-focused disease. And in the third phase, which followed and which we're still in, it became a disease whose geographic range kind of contracted again to the four corners of the Southwest and parts of California, but that also really consistently was causing a couple of human cases a year. And I think, Daniel, do you have those figures? The Centers for Disease Control keeps a count, and I know that we looked into it.
Yeah, well, so on average, starting from like 2000, so in the last 20 years, the average human cases is 7 cases per year. So,
Yeah,
yeah.
so an average of 7, some years more, some years far fewer, just 1 or 2. But what's unique about this third era is that humans re-enter the picture, but they're not getting infected to the same degree as wildlife, and there aren't epidemics that are as large as those among wildlife. And this brings us back to your question about the ferret, Victoria.
Mm.
So yeah, so in this kind of third era, or we can just say today in modern times, the species within the US that are most affected by plague are ground squirrels and prairie dogs, several different types. And in prairie dogs in particular, plague is extraordinarily deadly. When a plague outbreak hits a prairie dog population, it can kill up to 100% of the population, leaving, one or just a handful of members of the colony behind. And to explain this, it helps to know that prairie dogs are highly social animals. They are what ecologists call colonial animals. They live in colonies in which they work closely together, and that facilitates the spread of plague. I'm going to kind of take us on a slight detour back to Chinatown for a minute. One of the reasons why plague spread so readily in Chinatown was not only because people were living in such close quarters and in quarters that were occupied and infested with rats. It was also because when plague infects the lungs and people are living in close quarters, you can take the rat and the flea out of the equation, and people can transmit plague to each other. This is called pneumonic plague. They transmit it almost the same way that COVID-19 is spread, via the respiratory route. And some plague experts think that prairie dogs suffer such high mortality because they too might be able to spread plague via the respiratory route in their colonies. So today we have these horrible epizootics, again, among species like prairie dogs and ground squirrel species. And what you can kind of conclude is that if the prey is largely affected by an infectious disease, then the predators are going to feel the effects of that disease's spread as well. Way back, I think in the late 1920s, one scientist in the West looked at the outbreaks happening among prairie dogs, and he predicted that one type of ferret, the black-footed ferret, was likely to really suffer as a consequence of the spread of plague among prairie dogs. It preyed nearly exclusively on prairie dogs, and he predicted that the ferret would probably go extinct if no measures were taken to control plague. I guess the ferret didn't have a lot of defenders in the 30s and the 40s, because by the it's all but extinct in the US. And it was until sometime in the early 80s when scientists described to me that a colony of black-footed ferrets, just a small single isolated colony, was discovered. I believe it was in Wyoming. And so conservation efforts developed to try to conserve this ferret and reintroduce it back into the wild. But the moment one of these ferrets preyed on an infected prairie dog, the ferret would suffer as well. And so conservation efforts focused on the ferret have been thwarted by the persistence of plague in these prairie dogs. There have been some attempts that I know of, but don't know deeply about, to vaccinate prairie dogs in order to protect the ferrets. Of course, this would protect the prairie dogs too, but the prairie dogs have proven kind of more resilient. Even though they suffer terribly in the face of a plague epizootic, they also tend to bounce back. The few members who survive an epizootic will reproduce, or they will move to another colony and reproduce there. And so we see plague going in cycles of 5 to 10 years in prairie dog colonies. Among predators, it hasn't proven as epidemic as it does among these kind of more social colonial species. It's also interesting to know that plague is not as serious a disease for different types of species. It's very serious for prairie dogs, as I've mentioned. It's very serious for black-footed ferrets, but the predators that fall in the category of the cat species, the mountain lions, the bobcats, when they prey on an infected rodent, they suffer a high risk of serious disease and death. But the predators that are in the quote unquote dog family, like foxes and coyotes, they can prey on a plague infected rodent and be fine, have a mild illness or no illness at all. So it's interesting that this disease that started as a human urban disease has become a real problem among wildlife. And one estimate was that for plague to be causing the amount of death that disease ecologists are aware of, it's probably killing millions of animals every year.
And what about, there was some reporting that you did, so not only on the ecology of the West, which is fascinating, and fascinating the degree to which ecologists have a hard time getting their heads around the real impact, because so much of this happens unrecorded, unnoticed really. But that the other piece of the reporting that we weren't able to keep in the story just due to space constraints was about the effect that climate change may have on the resurgence of plague in the US or not, hopefully.
Yeah, so it's an interesting question. One of the things that I think was fascinating to both me and Daniel as we reported this piece was both that we have so much historical knowledge about plague, but it feels to me at least that it's not getting a ton of research attention. It was not terribly difficult for us to find the plague experts and for them to really bring us up to speed and spend a lot of time talking to us and informing us about the diseases past and the diseases present. But one of the other things that we were struck by is this disconnect between how serious it is in the wild, how relatively uncommon it is among people, and how as recently as the 1990s, scientists at the CDC were saying we really had to watch out because if plague found its way into an urban rodent population, we would be facing epidemics again. And this was pretty shocking to see this mentioned as recently as the 1990s. I should also mention as an aside that because plague is bacterial, if you catch it early, you can treat it with modern antibiotics. So it
So
isn't
it's not fatal to humans the way it was.
right, exactly. But this also means that some scientists have been thinking about what else might cause plague's range to kind of change or shift again. And one of the obvious factors is obviously change in the climate. Thinking back to that 1940 Surgeon General's report again for a moment, back then, those scientists surveying the West were shocked by how many different environments and landscapes plague was found in. But then, as I mentioned, in the third era of plague, its geographic range contracted. So the question now is the climate changing in ways that might cause its geographic range to change again? And it seems so far that this is really an open question. Scientists are still looking into this. They don't have any firm conclusions about it. It does seem that the answer depends on where you're looking and what part of either this country or the world you're talking about. For instance, I think in Central Asia, and plague is thought to have originated in Central Asia more than 2,000 years ago. It turns out there, I believe the prediction is that as winters get wetter, plague risk will increase.
Right.
Whereas here in the US, scientists have, in some studies, indicated that as summers get wetter, plague risk will increase.
I mean, that's because fleas tend to thrive in moist climates.
That is one of the reasons, yes, that some flea species do thrive in those conditions. But then, on the other hand, there's a lot of evidence that suggests that plague risk actually increases following drought conditions or in arid, dry conditions. And some have pointed out that if you look back at plague in the Middle Ages, what you see was bad outbreaks following years of drought. And there are different theories to explain this. Some are kind of more sociological, that, hungry people are more susceptible to disease. Hungry rats and thirsty rats move closer to humans and then may be in closer contact with humans and may provide more opportunities for disease to spread. But some of the more recent research, just looking simply at the rat and the flea vector, has indicated that when rats are affected by drought conditions, when they're hungry, when they are lacking nourishment, they start to weaken, they have to conserve energy. And one study that we read suggested that the rat that's conserving energy is actually spending less time self-grooming, less time pulling the fleas off of its body, and the flea population can proliferate. So this is one of these stories that seems like, things could go in any direction. One study, which I think is worth mentioning, looked just at plague in California, and showed that with the current climate trends, it seems likely that plague risk will actually decrease in the southern part of the state and increase in the northern part of the state and in the Sierras. So it's really anybody's best guess for now. But what we do know is that over 100 years, plagues kind of geography has already shifted. So it seems likely to shift again.
Continue to. And part of the reason we wanted to do this story and look back at this history is because it does inform us today, and super curious to hear what you've learned and who you talked to about what COVID or coronavirus might mean for wildlife and where we are in our understanding of its impact, or potential impact, I should say, on wildlands.
Yeah, so as we mentioned before, plague turned out to be a generalist. That meant that it could infect lots of different animal species, wild species, not just humans and not just rats. And very, very early research on COVID has looked at, and in fact, this one study in particular that we were struck by was just published last month. And in it, UC Davis researchers looked at a particular type of receptor that looks a lot like the receptor on our human cells that COVID-19, or that's the SARS-CoV-2 virus, binds to. This is the ACE2 receptor. It's on many of our cells, and a lot of mammalian species have receptors that closely resemble our ACE2 receptor. So their thinking was we should look at whether or how likely SARS-CoV-2 is to bind to the receptors in these other animals. And they looked at about 400 of them. And for several dozen, they predicted that coronavirus had a high or very high likelihood of being able to infect those species too. So the question then becomes, could COVID be a generalist? Could it find a reservoir among wild species anywhere in the world, or, because we're talking about plague here in the US? It turns out there weren't that many species, a species of deer and a species of muskrat in the US that were likely to be infected by coronavirus. But perhaps it only takes one, and this is a virus that is, of course, still evolving and likely to evolve. So, what the history of plague made us think about was a couple of things. One, could this coronavirus, turn out to be a generalist? Two, if we control its spread in humans, would we be missing a really critical opportunity to follow its spread in animals if we don't give that a lot of attention? Because this is what happened back in the early 1900s. After 1908, we breathed again such a sigh of relief that plague was controlled in the city of San Francisco. But little did people know that it had already spread across the bay into the ground squirrels of Contra Costa and Alameda counties, and from there all the way to what was called the plague line in the Rockies. So the other thing that comes to mind is that the way disease ecologists think of plague is as a disease in its ecological infancy. It's been here 120 years, and yet our prairie dogs still suffer a mortality rate of close to 100% in the worst epizootics. That's not the case in Central Asia, where the disease first evolved. And the thinking is quite simply that rodents and other animals there have had millennia to adapt to it. And our wild species here have only had 120 years, only 120 years. Right. So we probably have a very long road ahead of us with this new infectious disease. mean, this isn't to say that the story of plague is all caution and scary tales. I mean, the kind of good news about plague is we no longer live in an era of plague epidemics. We
Right.
live with a handful of human cases a year, but we do live again with possibly millions of cases among wildlife each year.
It's
So
exciting that the UC Davis, some of the folks there just jumped on this so quickly. You know,
it is.
it
It
just
is.
points to us being in a very different place.
Yeah,
And I am gonna take a look at some of the questions here now and
sure.
see, since I'm realizing we've been chatting away. Let's see. One reader suggests best introduction I know: the Black Death, the world's most devastating plague, a 24 lecture course by Dr. Dorsey Armstrong. Does that ring a bell to you?
No, I don't know it, but I'll be very curious to look into it. We probably should have watched it before writing this piece. Thank you for that.
Somebody else makes a comment. MC O'Connor says, remarkable lack of PPE on those public health workers dissecting rats.
It's shocking. It's shocking. And in fact, now we know that that is actually a great way to get plague. And it's entirely possible that some of those workers got plague back then. I don't recall reading about any cases like that. Do you, Daniel?
No, no.
No,
So
but you're
they
right.
may have just answered this next question from Jacqueline Turner. How did they check the bacillus in necropsied rats in the 1900s? How did they
Oh,
check for the bacillus? Excuse me.
you know, the very simple answer is that they took samples. So plague causes something called buboes in the human body. These are swellings in the areas where the lymph nodes are, and they show up in the groin and the armpits and sometimes elsewhere on the body. It turns out that plague causes buboes in rats as well. So they would take samples of tissue from the rats' buboes when they could find them, and they would look for them under the microscope. The bacillus had been identified, I believe, in the late 1880s or early 1890s, and it was known to have a very distinctive rod shape. And so they would take those samples, look at them under the microscope. I mean, this was painstaking work, and they did it in thousands and thousands of rats. I think in 1907-08, they collected, I have the number here, I think it was something on the order of 350,000 rats. And again, looked at every last one of them. That's a good question.
Yeah, yeah. A few people asked it actually.
Oh,
And then a few other people ask, can you speak a little bit more about a vaccine for plague in wildlife? And I'm curious, so if a vaccine works in prairie dogs, how transferable is that medicine to another rodent? Or is it, I'm really out of my depth here.
yeah, you know, actually, I don't know the answer to that. I don't know if the vaccine that's being developed for prairie dogs would work, say, for ground squirrels, which would be the other species that's experiencing the largest epizootics. I think that what I do know is that the vaccine is orally delivered, so it's delivered in bait, and the hope is that the prairie dogs will find it and consume it. I don't recall how effective it is. I recall how effective the human vaccine was. And the human vaccine was developed in the 1890s too, not long after the bacillus was identified. And it was actually proposed to be used in the first outbreak in San Francisco, but it was such a poor vaccine that offered so little protection and caused such bad side effects that people really resisted it, and it wasn't widely used. Um,
And
I
now,
and now there really isn't much interest in a human vaccine here. Most of the interest that I'm aware of is in vaccines for wild species. Again, some interest, like not
but
just
you're saying because we've developed antibiotics that
exactly,
treat it.
exactly.
I see.
Yeah, yeah.
Let's see. Steve, I'm going to mispronounce your last name, Steve, forgive me, but Ayala just says, very well reported, Elena and Daniel, thank you.
Thank
Um,
you.
Thank you.
uh, and he also says main sylvatic reservoir is wild rodents, which is less lethal to them. Although I'm not, perhaps that's a statement rather than a question. It doesn't sound like from the reporting that you've done that that's the case.
Um, well, the what we learned was that the it's true that the main sylvatic reservoir is wild rodents, but that actually encompasses a number of different species and rodent types. And it is, in fact, less lethal to species of rats than it is to prairie dogs. I've talked a lot about how mortality rates among prairie dogs are so high. The mortality rate is far lower among rats, and I hesitate to give you a figure because it will be an estimate, and I believe it was probably on the order of 50%, but it was enough, it was a high enough mortality rate that during the urban outbreaks of the early 20th century, some people would report seeing dead rats not long before the first cases of human plague turned up. But it wasn't like suddenly every rat in the city was dead the way that, plague can course through a prairie dog colony.
Yeah.
So yeah, so it's true. It is the case that plague is already less lethal. And one of the reasons is possibly that the disease has had more time to evolve in rats that have also crossed borders along with the disease. And so they've had a chance to evolve more resistance to it than, say, our native ground squirrels or prairie dogs.
We have an interesting question here from Beth Slatkin. Should we stop feeding our squirrel neighbors? Could we spread it back to them that way, meaning COVID?
Oh my goodness. I don't believe that squirrels, if Beth is talking about COVID, I don't believe that squirrels were high on the list of susceptible species, that is, species likely to be infected by COVID. Again, the ones that I remember from our part of the US were certain species of deer and muskrats. So I don't think we have to worry too much about squirrels. I do know that if you go to the zoo these days, if any Bay Nature subscribers go to the zoo, to any zoos that are open, there will be signs keeping visitors away, for instance, from the primates who have a very high likelihood of becoming infected with COVID-19. And, you know, the ones that are in the zoos here aren't in the wild here, but elsewhere in the world, there's probably a much greater worry about protecting primates from COVID-19.
Yeah, yeah.
As Beth meant plague, don't, yeah, she probably did. And I think that if you're in Yosemite, and oh, Daniel might want to mention this because there was a case, there are with some regularity cases of plague in Yosemite and elsewhere.
I'm so sorry. Somebody did ask that question and I went by it. I'll repeat it right now. Please also comment on current warnings about plague in Yosemite, Sequoia, Kings Canyon National Park, and then of course just last month in Tahoe.
Yeah, Daniel, do you want to speak to that? I know that you looked into that a bit.
Yeah, I mean, like you mentioned, Victoria, there was a plague case in South Lake Tahoe in August, and that was the first reported human case of plague in California in 5 years. The last reported human case was in Yosemite in 2015, which then two people tested positive, and both were treated and they recovered. Um, so yeah, I mean, I know, like, yeah, in Yosemite there are signs that warn people about plague and warn them that, not to touch the wildlife or don't feed them. So yeah, but in California specifically, yeah, that one in August was the first one in 5 years, and then prior to the 2015 cases, the last human case of plague in California was in 2006. So it's pretty low.
And yet the behaviors that result in these cases of plague are pretty consistent. Again, this kind of defines the modern era of plague. It's people who are coming in close contact with these reservoirs of the disease in the wild. So going back to the feeding squirrels questions, if you're camping, don't feed the squirrels. Those squirrels would be far more likely to have plague than plague in your backyard in an urban or highly developed suburban area. But the other cases that have cropped up in the last couple of decades have been in people who have like found a dead predator like a mountain lion or a bobcat and either explored it or were responsible in the cases of park staff for removing it and discarding it, cases of people getting it from their cats who got it from catching a mouse or a rat, and also campers and people coming in close contact with yeah, these, especially these rodent species in the wild.
And, Allie Judy here asks, I think, a natural question that I think I know the answer. She says, can the fleas, infected plague fleas, get on my dog and then onto me?
They can. The flea would have had to have bitten an infected rodent first and then not died while trying to feed on your dog. Um, in all likelihood, if the flea got on your dog first, it probably would just die of starvation before it jumped to you. But I mean, there's, yeah, I think a theoretical chance that if this all happens in close succession, an infected flea could move from a rodent to your dog to you.
And as you were saying before, in canine species in the wild, they tend not to be predators that are affected. So your dog is probably okay.
Yes, your
Yeah.
dog will be okay. Yeah.
Steve asks, and this we may be coming up on our last question here, or get your questions in if you have them. We're about to wind up. Parks are sometimes closed when an epizootic, or zootic, I should say, in squirrels is recognized. He cites Donner Lake State Park, and of course, areas were closed in Tahoe because I assume they're trying to get a handle on the prevalence of it in the wildlife.
Yeah, they're trying to determine how widespread it is, and they're also trying to simply reduce contact between humans and animals. And, this might be a good way to kind of go back to my answer to your first question, Victoria, about how the present is sort of different from the past. And I've emphasized a lot that in many ways it feels familiar. But one difference is that the event that gave us COVID-19, spillover, which happens when humans come in contact with wild species and their viruses jump into us and create disease in us, that spillover is happening more and more more and more commonly, more and more frequently than it used to, say 100 years ago. We have more contact. We have more humans living at the urban wildland interface. So we have more opportunities for spillover to happen. It also happens in places like wet markets and in places where people hunt and eat freshly killed meat from the wild. All of these kinds of events can increase the chances that a virus that is in a wild species can move into us. And so, yes, when those parks are closed, a big part of the reason is to reduce contact between us and the wild animals there.
So follow their requests.
Yeah,
Yes,
yeah.
the message. All right, we have come to the end of the hour. It went really fast. This was fascinating. And even though I edited this story and I've talked to you about it a lot, I learned more. So this is a really rich topic, both of you. And really thank you for all the work you've put into reporting this and condensing it to something that us lay people can digest. I really appreciate it.
Great. Well, thank you, Victoria. Thank you for inviting us to report on this story for you together. And thank you for having us on today. We appreciate it.
Yes, thank you.
Thank you. All right, take care, everybody. Thanks for joining us. Bye-bye.